Our talk is about the interaction between farming type, and this we consider natural regeneration and orchards, nutrient uptake, and plant material. This is clonal plants versus seedlings on fruit production and quality of strawberry tree plants. Strawberry tree is Arbutsunedo. It's a native species of Mediterranean climate, widely represented in southern Europe. In Portugal, we can find it from south to north of Portugal, associated to different species. Is resistance to forest fires, which make it quite important for forest programs, forestation due to forest fires in southern Europe. Is resistant to degraded soils, and the fruits are edible and can be consumed as fresh fruit. But main income is related to the production of a spirit called medronheira, which is the main income for forest owners. New products have appeared like yams, bonbons, chocolates, liquors, and dried fruits that can be incorporated in new products as cereals or yogurts. Due to the interest of forest producers in the economic valorization of these species, new orchards have been established using seedlings and clonal plants more productive. In this study, two farming types are going to be compared. Natural regeneration will be compared to orchards, and in these orchards it was established and trial where clonals were compared to seedlings with and without fertilization at moment of plantation. The experimental areas are located in center of and south of Portugal for natural regeneration, and for orchards they are located in center of region. Natural regeneration they have 25 years old, and orchards they have 7 years old. To know though, to identify the nutrient cycle soil plant and its relationship to fruit production, soil samples, organic layers and leaves were collected during spring to nutrients analysis, and fruits were harvested during autumn for culture analysis. At soil laboratory analysis, the different analyses were performed to identify different variables, pH, organic matter, and macro and micronutrients at soil samples, organic layers, leaves, and fruits. At chemistry laboratory, fruits were weighed. It was measured the diameter. It was identified different variables as total soluble solids, TSS, Briggs grade, acidity, and reduced and total sugars in order to evaluate fruit production and quality. Soils at Saint region are canby soils and lactose soils from shale rock. The pH is acid, very organic matter is 5%, very low level of phosphorus, and also very low level of calcium and magnesium, and a medium content of potassium. These conditions, the plants have soils show very low fertility. Soils in south of Portugal are alluvial soils from clay shale, where we can see here natural regeneration areas with neutral pH, very high level of organic matter, 19%, very low level of phosphorus, similar to the previous one, but with high content of calcium, magnesium, and very high content of potassium. This means that in these conditions, the soil fertility was higher compared to the previous one. At the organic layers show higher relevance on natural regeneration area, achieved to 12 centimeters over the soil. These graphs show the nutrients at the organic layers. We see that nutrients are more important as nitrogen, calcium, magnesium, followed by iron and manganese. At natural regeneration area, the total of organic matter, it was about 9.5 tons per hectare. And at our shards, we have a very inferior quantity of organic matter, about 2.9 tons per hectare. At natural regeneration, the ratio carbon nitrogen, it was 44, and at our shards 57. So we have much more important organic layers at natural regeneration, and important content of nitrogen, calcium, and followed by iron. These graphs shows the nutrients, macronutrients, and micronutrients on leaves and fruits. Leaves are shown in dark green and light green for natural regeneration and orchards, and fruits are red for natural regeneration and yellow for orchards. As we can see in this graph for macronutrients, we see that leaves showed same equal quantity at with as nitrogen, but concerning to potassium, we know that this is mobile in soil and plant, so it's quite similarly the quantity of potassium in leaves and fruits. As we saw before, we have very low level of phosphorus in soil, so we have also very low level of phosphorus in leaves and in fruits. Concerning to micronutrients, they are not mobile in soil and plant, so we have in leaves higher quantity of micronutrients compared to fruits. However, we can see here that iron content in fruit is higher in orchards compared to natural regeneration. This is linked to the acid soil observed in the orchards. These tables shows fruit production and nutrients export by fruits. Clonal show higher fruit production, as you can see here, compared with seedlings. So clonal plants export much more nutrients. In fruits, the distribution pattern shows this decreasing order: potassium, calcium, and magnesium. According to these results, clonal plants achieve to export an average of about 2 kilos per hectare of potassium, and concerning the micronutrients, this is about 11 grams per hectare. This is again associated with the orchard established in the first soil with a very acid pH, so with high content on iron. These graphs shows the orchard production and export of macronutrients. Concerning these treatments, as vegetal material seedlings noted as SE, and clonal plants as AL, and fertilization treatments without fertilization, the control noted with 0, with the fertilizer with these concentrations, with higher concentrations with phosphorus and potassium, noted as 133, and the control release fertilizer with similar concentration with this one, but added of magnesium and boron. As we can see in this graph, the lower production was observed with seedlings without fertilization, followed by clonal plants similar without fertilization. And then the higher production observed 7 years after plantation was observed with clonal plants fertilized, which achieve at over 1.5 tons per hectare. We can see also the most important nutrient extract, and that aligns with production, it was potassium, followed by calcium and magnesium, and very low levels of phosphorus and nitrogen. This is similar to the previous graph, but concerning to the export of micronutrients. In this case, we see that higher production again clonal plants and fertilized, and the high association between iron extraction and production. This, we think, again related with acid soils and its content on iron. This is a PCA analysis to study the soil-plant system. We can see that the factor 1 explains almost 29%, and it is linked to site index. Site index is related to plant vigor, and we can see here that plants show high plant vigor when they show higher organic dry matter at organic layers. And these cases, with higher content of organic layers, they show higher nitrogen content on organic layers and inferior ratio carbon-nitrogen. On the other hand, leaves show higher content with phosphorus and nitrogen. The second factor that explains the 23% show that higher content on organic carbon on soil samples are associated with or induce or promotes higher content of nitrogen in leaves. On the other hand, older plants, disease at natural regeneration, have leaves with high content with potassium and a lower content of nitrogen. This is a cluster analysis that confirms that fertilization is associated with production and higher extraction by fruits of potassium. On the other hand, we see that boron, the content of boron is associated to the type of vegetal material. Block factor is that explains less of the variability observed. This shows the effect of Orchard fertilization on fruit production and quality. 7 years old, this means this fertilization had been applied during plantation, and it was yet we can see that plants fertilized show a significantly higher production and with similar quality pattern concerning to bricks grade. This graph shows the interaction of fertilization and vegetal material on fruit production from 5 to 7 years old. 5 green, 6 years old at brown, and 7 years old at blue. As we can see, clonal plants are on the left and seedling plants on the right. Without fertilization noted it's 0 in both cases, and on the right of both cases the plants that were fertilized. We can see that fertilization improved the production for both cases, either clonals or seedlings. However, with clonal plants we obtain higher production and higher productivity. Moreover, with clone plants, we observed that the juvenility period, it was reduced. So they start production much more higher compared to seedlings. So we have production from 5, 6, and 7 years. Here we start production only at 7 years old plants. This graph shows the effect of fertilization and vegetal material on fruit quality. The vegetal material on the left side of the graphs and fertilization on the right side of the graphs. We can see here that clonal plants show inferior low values for fruit quality concerning bricks grade, acidity, reduced sugar, and total sugars. However, these values are not inferior, which is allowed for quality pattern. This means that higher production successfully observed with clonal plants means that it will be convenient to improve soil fertility in applying fertilizers or applying organic matter. As final considerations, we see that nutrients in leaves show this distribution, decreasing this distribution, nitrogen, calcium, and potassium. The extraction by fruits show this distribution, potassium, calcium, and magnesium. Our results show that it is relevant to promote and maintain the organic residues on soil and to select clones well adapted to the agroecological conditions. Clonal plants with great productive potential show a better response to fertilization. It's relevant to perform fertilization while planting, but also afterwards, in order to compensate the nutrients extracted by fruit production. At plantation, the fertilization with phosphorus and potassium improve fruit system development, and its effect on fruit production was noted even 7 years later, with 7 years old plants. As predictive features for future studies, we noted that it's important to define the most appropriate time for leaves harvesting to assess the crop nutritional status, establish the relationships among leaves nutrient content in fruits and production. Assess a suitable range for leaves nutrients to achieve the highest production potential and leaf reference values to optimize production and to support a fertilization program. And we would like to thank you for your attention.